Corals are marine invertebrates that are related to jellyfish and anemones. Many types of corals live in colonies of identical individual polyps. These corals help form reefs, such as the Great Barrier Reef. Today I'm going to be talking about timed coral mass spawning. One of the ways corals reproduce is they mass spawn by releasing eggs and sperm into the water at the same time in the hopes that the eggs and sperm meet each other and fertilize the egg. Coral mass spawning happens once a year during the night over the span of a couple weeks. Until recently, scientists thought that coral mass spawning was triggered by high water temperatures, but recent research has found that mass spawning is actually triggered by a rapid increase in sea surface temperature. So if this were a graph, if this was temp and this was time, researchers used to think that at this point that was when coral mass spawning was triggered, but actually what they've now realized is that during this change of rapid sea surface temperature, that is when coral mass spawning is triggered. The study, conducted over 16 years, found that there is no optimum temperature for spawning, with the same species spawning in different locations at different temperatures. For example, corals off the eastern side of Australia spawn when the water is 22 degrees Celsius. However, the same coral species in the Persian Gulf spawn when the water is 32 degrees Celsius. That is a 10 degrees Celsius different with the same species. Instead of using this high water temperature, in order to synchronize release of gametes and increase the probability of successful fertilization, the corals use the rapid change of temperature to time their reproduction. Having this knowledge will really help with dive tourism and the management of coral species. Processes like dredging that can really impact the early life stages of corals can be stopped when the corals spawn. However, as climate change continues and the timing of the rapid temperature increase changes, coral mass spawning timing can shift, affecting other species that use coral spawning as their own reproductive signal. Many other organisms besides corals time their spawning using different signals. Some use environmental cues like temperature to signal thermoreceptors, while others use chemoreceptors to sense pheromones. Pheromones are, as taken from the dictionary, a chemical substance that is usually produced by an animal and serves especially as a stimulus to other individuals of the same species, and results in one or more behavioral responses in animals of the same species. So, leads to behavioral response. They are used to trigger reproduction in many species, from polychaetes to fish. In Mozambique tilapia, pheromones in the urine of males boost hormone production and accelerates oocyte maturation in females. Pheromones are not the only type of signals organisms use to function. There are many different types of signals and receptors, like nociceptors, mechanoreceptors, photoreceptors, electroreceptors, and magnetoreceptors. All of these receptors receive signals and work together to share information with the organism. Nociceptors sense pain like when tissue damage occurs, and photoreceptors respond to light wavelengths so an organism can see. Mechanoreceptors respond to pressure and are used in hearing and orientation in animals such as fish. Electroreceptors detect electrical fields like the ampullae of Lorenzini in sharks. And finally, magnetoreceptors sense magnetic fields which are used by migrating birds. Every organism uses some of these different receptors to receive signals from their environment, just like the corals use thermoreceptors to sense the rapid increase in temperature. All these signals lead to responses in each organism that help it thrive in its environment and make up a fundamental process of all living organisms. If you would like to learn more about the coral mass spawning research, please check out Coral Mass Spawning Predicted by Rapid Seasonal Rise in Ocean Temperature research paper by Sally A. Keith et al., published May 11, 2016. Thank you very much.